Hi, my name is Narsha Haran. My matrix number 1805777. So, today I would like to talk about the study hall, which is about the storage disease, our galactocele ceramide ethyl. So I will talk about how to diagnose the disease. So the disease may be diagnosed by its characteristic grouping of sickle cell, which is multi-nucleated globoid cells, and the demyelination and degeneration and destruction of brain cells. Fluorescent substrate 12, which is N-methyl and 7-nitro-2-oxal 1,3-diazol coil and aminodipenoyl spingoils beta-D-galactoside, which is called NBD galactoserobroside, are these things were synthesized and used for detection of galactose-fibroside activity. The enzyme elimination using the substrate were found to be extremely sensitive, yielding unambiguous results. So that's all from my part. Thank you.
Hello, my name is Ikmal Shami bin Adam and my matrix number is 178325. So for the case study 4, we are given the disease in cattle, which is the crab disease, which is also known as galactocele ceramide. So the objective of this case study 4 is to identify what is galactocele ceramide in cattle about, then to understand the process of synthesis of galactoceramide, which is one part of the chapter in the lipid metabolism, and there is new veterinary terminologies that we learn here, which is sphingolipid, which mean the polar head group's control of carbohydrate units. So I will talk about the causes of this disease. It is actually disease from human, but somehow cattle also got it. So this crab disease or galactoceramide ceramide is a genetic disorder which inherit from the parents, which is you get from their parents in the gene. It is a genetic mutation and a permanent change in the DNA sequences that make up a certain gene. So the mutation affect the message that the gene sends to the cells in the body. The gene for crab disease can be found on the chromosome 14. Then the abnormal genes result in a shortage of an important enzyme that body needs called galactosyl ceramidase, which is GALC. So a defect or deficiency in GALC, which is the main cause of the scrap disease. So the body needs the substance to make the myelin, the material that surround and protect the nerve fiber. Without it, myelin breakdowns, brain cells die, and the nerve in the brain and the other body areas do not work properly. So I think that's all from me for this case study 4. Thank you.
Hi, my name is Mary Laura Coming and my matrix number is 178685. So for today, I'm going to talk about storage disease in cattle named galactocele ceramide. So the objective of our case study is to identify what is galactocele ceramide in cattle And the second one is to understand the process of synthesis of galactosylceramide, which is one of the part in the chapter of lipid metabolism. So one of the new veterinary terminologies that we found is sphingolipid. Polar head, which means polar head groups consist of carbohydrate units. So what is galactocerebrosidase? A galactocerebrosidase or galactosylceramide is a type of cerebroside consisting of a ceramide with a galactose residue at the one hydroxyl at the one hydroxyl moiety. Galactocerebrosidosis is known as colloid cell leukodystrophy, galactosylceramide lipidosis, or Krebs disease. The galactose is cleaved by galactosylceramide. Galactocerebroside is a marker for oligodendrocytes in the brain, whether or not they form myelin. Krebs is a degenerative disorder that affects the nervous system. It is caused by the shortage or deficiency of an enzyme called galactosylceramidase. This enzyme deficiency impairs the growth and maintenance of myelin, the protective covering around certain nerve cells. Nerve cells that ensures the rapid transmission of nerve impulses. Krebs disease is part of a group disorder known as leukodystrophies, which results from the loss of myelin or demyelination. This disorder is also characterized by the abnormal presence of globoid cells, which are globe-shaped cells that usually have more than one nucleus. That's all for my part. Thank you.
Hi, my name is Selvi and my matrix number is 178830. Today I'm going to discuss about galactocerebroside, which is our last activity. To begin with, let me start off with the objectives of our case study. The first one is to identify what is galactoceramide in cattle about. The second one is to understand the process of synthesis of galactosylceramide, which is one of the part of the chapters in lipid metabolism. And the difficult or new veterinary terminologies that are found in this topic is sphingolipid, which means polar head groups or consists of carbohydrate units. So let me begin by discussing what is galactocerebroside. Galactocerebroside or galactosylceramide is a type of cerebroside consists of ceramide, which is a galactose residue at 1 hydroxyl moiety. Galactocerebrosidosis is also known as globoid cell leukodystrophy, galactoceramide lipidosis, or Krebs disease. The galactose is cleaved by galactosylceramidase. Galactocerebrosidase is a marker for oligodendrocytes in the brain, whether or not they form the myelin. Krebs is a degenerative disorder that affects the nervous system. It is caused by the shortage or deficiency of an enzyme called galactosylceramidase. This enzyme deficiency impacts the growth and maintenance of myelin, the protective covering around a certain nerve cell that ensures the rapid transmission of the nerve impulses. That's all for my part. Thank you.
Hi, I'm Shazwani Ahmad with matrix number 178014. So today I'm gonna discuss about biochemical process in galactosylceramide disease, which is an inherited disease in cattle. So for biosynthesis of ceramide and acylphenidosine, the biosynthesis of ceramide occurs in 4 reactions. From the precursors palmitoyls OA and serine. First, 3-ketosphingene synthase, which is serine palmitoyl transferase, pyridoxal phosphate dependent enzyme. catalyze the condensation of palmitoyl-CoA with serine yielding 3-ketosphingene, which is pyridoxal phosphate dependent reaction. Next, 3-ketosphingene reductase catalyze the NADPH dependent reduction of 3-ketosphingenine's keto group to form sphingenine, which is called dihydrosphingosid. Next, dihydroceramide is formed by transfer of an acyl group from an acyl-CoA to the sphingene's 2-amino group, forming an amide bond. And later, dehydrogenase converts dihydroceramide to ceramide by an FAD-dependent oxidation reaction. So, for the biosynthesis of galactose in ceramide, it is synthesized from ceramide by addition of a glycosyl unit from the corresponding UDP hexose. Galactocerebroside is common component of prelipids. Glucocerebroside is catalyzed by UDP glucoceramide glycosyl transferase, which is glucosylceramide synthase. Using that, and ceramide is formed. Ceramide is catalyzed by UDP galactoceramide, which is glycosyl transferase, and galactocerebroside is formed. Galactocerebroside is lysosomally degraded by a series of enzymatically mediated hydrolytic reactions. These reactions are catalyzed at the lipid-water interface by soluble enzymes. That's all for me. Thank you.
My name is Limini and my machine number is 178225. Now I will talk about the last case study, which is galactoceramide in cattle. I will talk about its cause and consequences and sign and symptom for the repeat steroid disease in cattle. Galactocerebrosidiosis is a member inherited sphingorhabdosis that results from the deficiency in beta-D-galactocerebrosidase. Beta-D-galactocerebrosidase is known as galactoceramide activity also. The substrate galactocerebrosidase is a constituent of myelin and another metabolite of myelin turnover of oxytocin accumulate. Cytosine is highly cytotoxic to oligodendroglial and thought to be the primary metabolite involved in pathogenesis of the disease. Consideration to the build-out of this toxic metabolite create disease result. It is pathologically characterized by bilateral symmetry, demyelination of white matter of the brain, spinal cord, spinal nerve roots, and peripheral nerves by the accumulation of white cells, predominantly perivascularly. For its signs and symptoms, initial signs and symptoms typically include irritability, muscle weakness, feeding difficulty, episode of fever without any sign of infection, stiff posture, and slow mental and physical development. As the disease progresses, muscle continue to weaken, affecting cattle ability to move, chew, swallow, and breathe. Affected cattle also experience vision loss and seizures. Visual deficit and blindness may result late in the disease, but ophthalmoscopy lesions have not been observed. Visual problems and walking difficulty are the most common initial symptoms in this form of the disorder. A progressive diffuse neurology syndrome develops at younger age, and they occurs within a few months. Okay, that's all from me. Thank you.